Good morning, children.
Good morning, ma'am.
Morning, ma'am.
So, hope you have no doubts regarding prophase 1 of meiosis. Any doubts regarding prophase 1? The 5 stages are clear. So, now let us proceed to the remaining stages. So, out of that PMAT, prophase 1 is over. So, we'll discuss about the remaining stages coming under meiosis. Okay, Sharon, no problem. So after completing prophase 1, we'll proceed to metaphase 1. As I already told you, in metaphase 1, you can find the chromosomes getting arranged at the, with the help of the spindle fibers arising from the centrioles. They'll get attached. These microtubules, which will be forming spindle fibers. They'll come and attach to the kinetochores of these homologous chromosomes. And bivalent chromosomes, they'll get arranged at the equatorial plate. So bivalent chromosomes will be getting arranged at the equatorial plane. Once again, I will repeat. So in metaphase 1, spindle fibers will be formed. Microtubules will form spindle fibers. And those spindle fibers will be getting attached to the kinetochores of sister chromatids in the homologous chromosomes. And these bivalents means these homologous chromosomes will get arranged at the equatorial plane. Then in anaphase 1, what you will find, these homologous chromosomes, not the centromere, is getting splitted. Like in mitosis, centromere will get split and the sister chromatids will move towards the opposite poles. But in anaphase 1, coming under meiotic 1 division, it's not the centromere which is getting split, but the homologous chromosomes will get separated and they will be moving towards the opposite poles. So these are the homologous chromosomes. So they will get separated and you can find them moving towards the opposite poles. Whereas the sister chromatids will remain attached at the centromere. Here the centromere will not get split. Now telophase 1. In telophase 1, the nuclear membrane and nucleolus will start to reappear. Then chromosomes will undergo some extent of dispersion. They will not get completely coiled, or they will not get the completely extended state as in the case of interphase, but definitely they will undergo some amount of dispersion. The reason is why they are not completely getting extended or getting coiled structure, because this particular stage will be followed by meiotic 2 division. But in between meiotic 1 and 2 divisions, there is a small interval, there is a small gap. You call it as interkinesis. Don't get confused between interphase. Interphase is the preparatory phase before the M phase. In cell cycle, we learned about M phase and interphase, which constitutes 95% of the cell cycle and which is subdivided into G1, G2, S phase. But interkinesis is the gap between two consecutive M1, meiotic 1 and meiotic 2 divisions. Now moving on to meiosis 2. So meiosis 2 also consists of the different 4 stages: prophase, metaphase, anaphase and telophase. It is resembling mitosis. Why we say it is resembling mitosis? Because in meiotic 1 division, a cell having for example, if you take a cell having 46 chromosomes, by the end of meiotic 1 division, that is after the telophase, we'll be getting 2 daughter cells, each having half the number of chromosomes. If 46 chromosomes were there in the parent cell, the daughter cells will be having 23 chromosomes each. That was in meiotic 1 division. Generally, you call meiosis as reductional division. That is clearly proved in the first division of meiosis. But meiosis 2, that is same as mitosis. Here the chromosome number will remain the same, because at the end of At the end of meiotic 1 division, when meiosis 1 is completed, just imagine a cell having 46 chromosomes underwent meiotic 1 division. You will be having 2 daughter cells, each having 23 chromosomes each. Now, these 2 cells will be entering meiotic 2 stage, and each one will get divided into 2 daughter cells. Altogether, 4 daughter cells will be produced at the end of meiosis. Meiotic 1 and 2 together will result in the production of 4 daughter cells. Here you can find in meiotic 1, you found reductional division took place. The 46 chromosomes got changed to 23 chromosomes in the first phase, whereas in the second phase, the chromosome number remains the same in the daughter cells also. That is why we say it is resembling mitosis. Any doubt? Any doubt? I'm not getting your response. Any doubt, children?
No, ma'am.
Okay.
No, ma'am.
So, meiosis 2 is again subdivided into prophase 2, metaphase 2, anaphase 2, and telophase 2. So, one by one, we'll see the stages. Earlier, what confusion you had is I displayed one picture, because assuming that the both cells or both daughter cells, the same process going on. So, to avoid the confusion, I have shown both daughter cells. So in prophase 2, now you know why 2 daughter cells are shown. Because at the end of first meiotic division, you get 2 daughter cells. Dyad, we get dyad of cells, that is 2 daughter cells. Each of them will be entering into prophase 2. As in any other case, prophase stage is characterized by the condensation of chromosomes. Chromosomes become short, compact, and the nuclear membrane will be disappearing as prophase stage proceeds. Okay, 2 cells, both the daughter cells undergoing prophase 2. Now next is metaphase 2. Both daughter cells, 2 daughter cells are formed, both will be undergoing metaphase 2 stage. And metaphase 2 stage, as I told you, M for middle, chromosomes will get aligned at the equatorial plane. How do they get aligned? With the help of spindle fibers. Where are these spindle fibers attached? These are attached to the kinetochores of sister chromatids. These are attached to the kinetochores of sister chromatids, and these are the spindle fibers, and they will align the chromosomes at the equatorial plate. In metaphase 2, once again, chromosomes will get aligned at the equatorial plate with the help of spindle fibers. How are the spindle fibers attached? Spindle fibers are arising from the centriole, and they will get aligned at the equatorial. The chromosomes will be aligned at the equatorial plane, and these spindle fibers will be attached to the disc-shaped structures known as kinetochores present close to the centromere. Anaphase 2. 2 daughter cells are there. In both you can see the chromosomes, how they are splitting. Just like in mitosis, the chromosomes will split at the region of centromere. Centromeres will split and the sister chromatids will be moving towards the opposite poles. Very important point. In mitosis also the same thing is happening during anaphase. Centromere will split, separating the sister chromatids. Sister chromatids will be moving towards the opposite poles. Whereas in meiosis 1, centromere and sister chromatids were remaining intact, but the homologous chromosomes got separated. And here the centromere will get split and the sister chromatids will be moving towards the opposite poles. And then what happens? Telophase 2 begins. And in telophase 2, the chromosomes will decondense. It will decondense because again it will go back to the coil stage, nuclear membrane and nucleolus will reappear, and this will be followed by cytokinesis resulting in the formation of 4 haploid cells. See this 2 daughter cells. These are the 2 daughter cells formed at the end of telophase, anaphase 2. And once it is completed, each cell will be dividing into two daughter cells. So altogether we will get four daughter cells, 1, 2, 3 and 4. But by mitosis we get just two daughter cells, and by the end of meiosis 1 also you will be getting only two daughter cells, and these two daughter cells produced as a result of meiosis 1 will enter into meiosis 2. Each daughter cell will undergo division and form two daughter cells by the end of telophase. Here you can find this will be getting separated like this, and each one will be forming two daughter cells each. Clear, children? Any doubt? Yes or no?
No, ma'am.
Okay. Then significance of meiosis. Meiosis results in the reduction of chromosome number by half. Whereas in mitosis, chromosome number is maintained the same. If parent cell is having 46 chromosomes, daughter cell also will be having 46 chromosomes each. But in meiosis, if parent is having 46, exactly half the number will be found in daughter cells. So this will help to conserve the chromosome number of species from generation to generation. That is the significance. It will help in maintaining the chromosome number stable. It increases genetic variability in sexually reproducing organisms, because meiosis is a method of production of gametes. Division by means of gametes are formed. When these gametes are fused, it will promote the genetic variability. Thus, variation can lead to, that you have learned in 10th standard, accumulation of variations can slowly lead to the formation of new species, or you can say it can lead to evolution. Two main points: it will reduce the chromosome number to half, so that upon fertilization the zygote will retain the same chromosome number. It will help in maintaining the chromosome number of the species generation after generation. Second point, it will promote genetic variability in sexually reproducing organisms. Mitosis means identical, similar cells are formed, but meiosis can lead to genetic variability. See here in nutshell, you can see all the stages. So in prophase 1, pairing of the homologous chromosomes taking place. In metaphase, the homologous chromosomes are getting aligned at the equatorial plane. And in anaphase 1, centromere is not splitting. Sister chromatids are not getting separated, but the homologous chromosomes will get separated and they will form two daughter cells. And if you compare the number of chromosomes, here we had 4 chromosomes. 1, 2, 3 and 4. 4 chromosomes. But the daughter cells are having only 2 chromosomes each. So the chromosome number is reduced to half in daughter cells. And then each of these cells will enter into prophase 2 stage. So condensation of the chromosomes will take place. Again in metaphase, they will get aligned at the equatorial plane. And anaphase 2, not the anaphase 1. Changes taking place in anaphase 1 and anaphase 2. In anaphase 1, centromere is getting split, and they are moving to, in anaphase 1, the homologous chromosomes are getting split and they are moving to the opposite poles. But in anaphase 2, centromere splits and sister chromatids are moving towards the opposite poles. And here we started with 2 chromosomes, and the daughter cells also will be having 2 chromosomes each. That is why we call it as or we say that it is similar to mitosis. So by the end, so in altogether as a whole, if you take, the cell started having 4 chromosomes,
Many organisms pass their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have two copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have one copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are two cell division events during meiosis. The first division, meiosis I, results in two unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis II, results in 4 unique haploid cells that only have one copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase I. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis I. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase I. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis I is called metaphase I. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase I. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis I are telophase I and cytokinesis. Here, the cell divides into 2 daughter cells. Each of these 2 cells now undergo meiosis II. Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase II, the nuclear membrane reforms and the cytoplasm is divided into the 2 haploid daughter cells. This division is called cytokinesis. Since meiosis II began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Clear? Any doubts remaining? Any doubts you have? Yes or no? Have any doubts? It is clear now. And what is the major difference between the anaphase 1 and 2 stages of meiosis? There is one major difference between anaphase 1 and anaphase 2 of meiosis. What is that? Malavika.
In anaphase 1, homologous chromosomes get separate. In anaphase 2, sister chromatids separate.
Centromere
centromere.
splits and sister chromatids are getting separated and moving towards the opposite poles. Now, meiosis and mitosis are completed. Now, can you summarize the differences between these two types of division? Whatever points you know, so more we will be discussing. So, whatever you have understood, Vidur, just tell me one point of difference between meiosis and mitosis. Any one difference? Where does it take place? What for it is taking place? Or how many cells are being formed? So many points are there.
In mitosis, there will be chromosome numbers equal to the parent cell. And in meiosis, chromosome number will be half of the number of parents.
The cells formed will have the same number of chromosomes as the parent cells in case of mitosis, whereas in meiosis, the chromosome number will be reduced to half in daughter cells. So you call mitosis as equational division and meiosis as a reductional division. So one point is clear. Sania, another point.
In mitosis, 2 cells are formed, whereas in meiosis, 4 daughter cells will be formed.
So 2 daughter cells are formed as a result of mitosis and 4 daughter cells are formed as a result of meiosis. Then, what is the other difference, Gayatri? Where it is taking place, mitosis?
Mitosis takes place in body cells and meiosis takes place in germ cells.
For the formation of gametes. Any other differences? Any other differences? Evelyn? Evelyn, all the differences covered? Yes.
Mitosis is having 4 distinct phases.
That even in meiosis it is there, no? Mitosis involves only one division, means only one stage. Whereas in meiosis, 2 divisions, meiosis 1 and meiosis 2 are involved. Okay, a few more differences we'll see, and whatever points you've said, plus some more points you can see now. Can you see the screen? Children, can you see the screen?
Yes, ma'am.
Yes, ma'am.
Net?
Most breathtaking processes that helps the cell to not only grow but also divide. We know that a cell cycle has 2 major phases. One helps in the preparation of the cell for the division. Can you name the preparatory phase of the cell cycle? That's the right answer. It's called the interphase. And moving to the other phase, it carries out the scission of the cell. And what is that phase called? Correct. It's the M phase, where M stands for either mitosis or meiosis. And how does the cell decide whether to undergo mitosis or meiosis? Well, that depends on various factors and the type of cell undergoing division. Keep your eyes glued to the video to spot the differences between mitosis and meiosis. Let me help you with the first difference between the two processes. Mitosis occurs in somatic or body cells, while meiosis occurs exclusively in the reproductive cells. That means mitosis, occurring irrespective of whether the individual is a male or a female, gives rise to somatic cells, which are exact copies of the parent cells. On the other hand, meiosis occurs in males and females, giving rise to haploid sperm and egg cells respectively. The process of meiosis, however, has different names in both the sexes. Yes, due to the different cells produced as an end result, meiosis is also referred to as oogenesis, that is the process of ovum production in females, and spermatogenesis that is the sperm production process in males. Another major difference is that mitosis is used as a mode of asexual reproduction in lower organisms, while meiosis helps in forming gametes for sexual reproduction. Now, let's move ahead with the next difference. Have a look at these images. Observing this image, we can easily notice the most basic differences between the two. Let's begin noting down each difference individually. The first on the list is that mitosis has only one cell division, while meiosis involves two successive cell divisions. That simply means mitosis is a single-step division, while meiosis is a two-step division. And this results in meiosis having two phases, namely meiosis 1 and meiosis 2. Can you note down the second difference in the list? Correct. Mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells. And is there something remarkable that we haven't discussed yet? That's right. Now let's move on to the next major difference. This difference is based on the chromosomal number of the daughter cells. In mitosis, the daughter cells are diploid with 2 sets of chromosomes, just like the parent cell, while during meiosis, the daughter cells produced are haploid with only 1 set of chromosome. These daughter cells are called gametes, have exactly half the number of chromosomes from the parent cells. The former difference also gives cue about one more difference between the two. What could that be? Well, the image clearly depicts that all the 4 daughter cells produced by meiosis are genetically different from each other, while the 2 daughter cells that are produced by mitosis are genetically similar to each other. Now let's move on to the next difference. But before that, can you tell me how many stages of division does the mitosis process have? Correct. It has 4 stages of division. And is it the same for meiosis as well? The answer is a definite no. Since meiosis involves 2 rounds of division, that is, meiosis 1 and meiosis 2, the process has a total of 8 stages of division. Now moving on to the next difference, this point has been studied by us in the lower grades as well. We have studied that mitosis occurs in all organisms except viruses. And which organisms can have meiosis? Meiosis occurs only in plants, animals, and fungi. Now, let's dive deeper and compare the stages of mitosis and meiosis to explore some more differences between them. The first stage of meiosis 1, that is prophase 1, is very long, consisting of 5 substages. On the other hand, prophase of mitosis is comparatively shorter than prophase 1. Crossing over does not occur in prophase, while definitely occurs in prophase 1. Independent assortment is technically absent in the metaphase of mitosis, while it occurs in metaphase 1. The separation of chromatids takes place during anaphase, and on the other hand, the separation of homologous chromosomes takes place during anaphase 1, and the separation of sister chromatids during anaphase 2. So, these were the major differences between mitosis and meiosis. Let's summarize them in a single table. Before signing off, tell me one thing. Have you ever wondered what important tasks do these 2 processes perform in our body? Well, mitosis helps in the healing and repairing of the cells and tissues of our body. It's also responsible for the growth and the development of the body. Meiosis, however, helps exclusively in producing gametes. This ensures genetic diversity. And with this, we come to an end of the concept of cell cycle.
You can take a screenshot of this slide. Okay, the major differences. Have you? Is it clear, children, now? I think all the doubts are cleared. The different stages, differences. Any doubts still remaining? Clear? Yes, Steffi?
Yes, ma'am.
Okay. So we'll wind up. So come prepared. This chapter, I think we have completed the different stages. So next day when you come prepared for a test for this chapter. Yes, only this chapter, very small chapter, no? So come prepared for a test. It can be oral test or written. Prepare for both. Okay, thank you, children. See you next class.